Okay, so wheat is the most important crop in the world. And I'm not saying it is just because of production, but mainly because of the distribution of this cultivar. So you can see that it's growing all over. So every day there is some wheat is being harvested somewhere else in the world. And this is very important for food security. But it wasn't always like that, right? If we look at wheat evolution, so we know it's originated from the Fertile Crescent, and also the evolution is quite complex. I won't go into that into too many details, but just to show you that it's a hybridization of two diploid species, wild species, that gave rise to wild MR wheat, Triticum dicocoides. And here domestication happens, and this is durum wheat that today is being used for pasta. And then another hybridization with another diploid species and gave rise to hexaploid wheat, which is the 95% of the wheat that is being consumed today. I would like you to notice that if you look at the genetic diversity, it's going down because of those bottleneck of domestication and polyploidization. So if my research is focusing on wild wheat as a way to identify beneficial allele from wild wheat and to improve cultivated wheat. And the idea is to use this genetic variation that exists in wild wheat. We are looking into yield and quality and also heat tolerance. Wild wheat is growing in warmer places in the Fertile Crescent. So, and it is shown to have genes responsible for heat tolerance, which is becoming very important due to the global warm. And our strategy is to develop parental population for genetic mapping and gene discovery. And here you can see the distribution of wild MO wheat, and also the size of the circle indicates the gene diversity of the subpopulations. So if you're working on the biparental population, you have to choose carefully because the amount of parents that you can choose is limited. And here I collaborated with Professor Jan Dvořák that developed one of the population from Turkey, from the region that is assumed to be the origin of domestication. And another two population I chose from the Israeli subpopulations. And the Israeli subpopulation are very interesting. You look here at the population structure, and you can see that there is an outlier clade here. This is sometimes called as the Judaicum variant. And there are also northern population and southern population. So one population we chose from the northern dicocoides, and the main population is from this Judaicum variant. And this is how it looks like in Israel, in the Zavitan Nature Reserve, where this line was collected. And for me, this is more exciting than polar bear, but just me. So you can see the Sea of Galilee, the Zavitan Nature Reserve, and wild wheat growing. So it's, I think it's cool that we can collect it and have the genome. So this is what we hope for. And so I'm focusing on yield, and grain size is one of the yield components. And you can see here the domesticated wheat, the variation in grain size is not that great. So over the last 50 years, not much had happened in the grain dimension in domesticated wheat. But if you look at the variation, the phenotypic variation that exists between wild MR wheat accessions, so you can see the accession that is from Turkey, the origin of domestication, the small grains, and the grains from the Zavitan accession, the main population that we developed. And you can see the variation, the phenotypic variation. And of course, as a geneticist, I'm interested to know what are the genes responsible for this variation. And to date, none of the gene that is responsible for this natural allelic variation is known. So it's a valid biological question. And when you go deep into those grain size between wild and domesticated wheat, there is interesting things going on. So in wild wheat, in the spikelet, which is the distribution unit, usually you have two grains. One is lower and one is upper. And as you can see here, the upper grains are bigger than the lower grains. And in domesticated wheat, these differences disappeared. So this has happened during domestication. And what is nice about it, that the upper grains germinate readily in wild emmer wheat, while the lower grains show seed dormancy. And this is a known biological mechanism of wild species that they remain part of their seeds as a gene bank if a catastrophe is happening. So next year you will have seeds that can germinate. And of course, under domestication or agricultural condition, this trait is unwanted, and this is why it's disappeared. So we are going after this trait, and actually now we identified a major QTL that is responsible for those big differences. And so to move forward and to map those interesting traits, we developed a real population in F6. Now it's F8. You can see there's Zavitan, the dicocoides, wild wheat, which is very robust. The other parent is the Italian accession Svevo, which is growing very nicely here, producing high quality grains. And you can see the field experiment that we set up last year. This year we repeat this experiment for the second year. It's still just germinating. And those are just first results from that shows that positive transgressive segregation for grain size. You can see the real population. And this is Svevo, which has wide seeds but short. And Zavitan that has long seeds but narrower than Svevo. And what is positive transgressive segregation is those accessions that have both wide and long seeds, so they have higher grain weight. Of course, we have to see if there are not any trade-offs, but this is something that has a potential for yield increase, and this is something that we are going after. So I hope that at that point I persuade you that we have interesting phenotypic variation that is interesting to study. So what about the genetics, about the genotypic part? So we invested in the Svevo-Zavitan population quite heavily. We used the 90K chip to genotype the reals and got more than 14,000 markers, SNP EST-based markers, so those are genes mainly. And we also used Energene. Energene provided us with a GBS map that has more than 900,000 markers. And this is how it looks like, the wheat chromosomes, the centimorgans, and you can see that it's very dense, tens of thousands of markers per chromosome. And a work done by Macaferi et al. Recently, a great paper, shows consensus mapping. And you can see the different maps aligned to the hexaploid wheat chromosome, the only chromosome that was sequenced. So you can see in our maps, the Svevo and Zavitan, which has that shows the best resolution and coverage. And this shouldn't surprise, because as I was mentioning earlier, the genetic variation that exists in wild wheat is higher. And this is why we have resolution in regions that are fixed in domesticated wheat. So that's genetics, that's the beauty of it. And so when you compare the markers, in the x-axis is the genetic distances in wheat to the genome of barley, you can study collinearity because the gene order is preserved. And you can get a genome view of tetraploid wheat with this mapping population. So you can see small inversions that happened after the wheat and barley divergence, right? So this is known inversion that we can see them. And also inversions and rearrangement that happened in chromosome 4A after the A and B genome diversion. So we can have, and this is important, of course, if you want to go after a QTL, it's important to know where you are at the map, because as you can see, the line is not straight. And why it's not straight? Because of the reason of low recombination in the centromere. So for instance, if you have a QTL here, maybe map-based cloning is not going to be the best approach. If you have a QTL here, you don't have a problem with resolution, because you have a lot of recombination, and you can move on with map-based cloning. So we were quite happy with this genomic view and the map, and decided with Energin to move forward and to sequence the wild wheat genome. And this is a project that we initiated. And the strategy is very simple. We use Energin used short read Illumina sequencing data that they build scaffolds out of it and anchor the scaffold to the genetic map. And as I showed you before, 1 million markers should be quite enough to anchor most of the scaffold, because the resolution is very high. And this is an ongoing project. And so, you know, I'm not a bioinformatician, so numbers not always impress me. So I wanted to validate the GBS data, because we are putting heavy investment on this data. So how to validate? We validate using biology. So I'm going back to the grain size differences. So using those two accession and the population derived from the cross between this accession and this accession, the Turkish one, and the Durum from the 50s, population developed by Jan Dvořák from UC Davis. We identify a QTL for grain weight on chromosome 5AL. And this mapping was done, and the flanking markers are SNP1 and SNP6 that are defining a 70 centimorgan region on chromosome 5AL. And we validated this QTL using near-isogenic lines. You can see the near-isogenic lines that show higher 1,000 grain weight compared to the Langdon, compared to the domesticated wheat control. The shape of the spikes, because it's a near-isogenic line, is identical to the control. But still, you get higher weight and higher volume. Those are 500 seeds. We counted each. And what is interesting, that is this QTL is actually a meta-QTL. What is a meta-QTL? It's a QTL that was identified in other populations. So this gives our more, so this encourages that this is a significant QTL. You see here, this is the meta-QTL 7 that Ling et al. In their Tritico-Murartu sequencing paper described. Tritico-Murartu is the donor of the A genome of wheat. So this is why it's important for us, because we can get sequence data from this region. So this is what we did. This is the region. Those are the two near-isogenic lines, and the overlapping region is the region, the critical region that is responsible for the grain weight QTL. And when you look at the Tritico-Murartu genome, we identify, or the authors here identified 6 scaffolds, which corresponds to about 0.5 mega of base pairs. So when what we did here is a simple comparison. We identified this region in our GBS map, and there was 573 GBS markers and 43 SNP-based markers. And we did blast against the Tritico-Murartu genome. And this enabled us to identify 124 scaffolds, which is 10 mega of sequence data. And this is 20-fold improvement from the previous analysis. And in this region, there are around 150 genes. And this is enabling us now to use RNA-seq approach and exome capture approach to identify, to compare between wild and domesticated alleles, and give us, and maybe to identify the causative genes for this important QTL. By the way, we did this analysis for the whole genome. We took our GBS map, aligned it to the Triticum-Urartu genome, and we were able to anchor 40%, 2 giga, of the Triticum-Urartu scaffolds, even though the N50 of the Urartu scaffolds is around 60 kb. So 40% alignment of the genome is quite good, and this is, for me, this is a proof of concept that this genome sequencing can work, especially if the new technologies and energy abilities are should supply with higher N50 scaffolds. So the higher N50 the scaffold, there is a even better chance to anchor those scaffolds to the genetic map. And this is it. Oh, sorry. So the markers from this region, yeah. So we have markers now, but we also need to validate them. And this is the graphical genotypes of the secondary AB reels that we are using for the QTL region. And you can see that those markers, they make genetic, there is a genetic logic behind this because they are mapping, those are the original near-isogenic lines. And you can see that we have a lot of recombinations, and the recombinations make sense, and we can identify the introgression in each of these lines. And of course, what we are doing now is characterizing it in the field, and hopefully we'll be able to identify the causative genes by using this approach. And I would like to thank my collaborators in this project, Abraham Korol from the University of Haifa, helped with the genetic map, Jan Dvořák, and Chad Jorgensen from UC Davis, Zvi Pelek from the Hebrew University, and Hansela from Tel Aviv University, and of course Energene, and also the funding agencies, the Israeli Science Foundation, the Marie Curie Reintegration Grants. And I would like to thank you.
First of all, congratulations. Nice piece of work.
Thank you.
Just out of curiosity, to what extent, if any, is this 1,000 kernel weight related to phenology?
Okay, so this region is close to VRN1, but we made sure that VRN1 is not in this region. So actually, the near-isogenic lines and controls are flowering at the same time, okay. And this is why the phenology has no effect on this QTL.
Ah, very nice. So it's a QTL for yield per se mainly.
Right, right. And it's also not, VRN1 is also close to the Q locus, which affect domestication traits, and the Q locus is out of this region.
Have you looked at roots of these plants?
No. But
thank you.
Yeah, but the thing that, the fact that this locus is adjacent to the Q locus, maybe it's the explanation why this QTL left outside during domestication, because the selection moved during fixing the Q locus and not to the
my
QTL.
question, I'm interested in the physiology behind it. So I was wondering if you could share some thoughts about what's behind this. In terms of, have you looked number of endosperm cells or size of the single endosperm cells?
Not yet. We would like to do it with the new isogenic lines, that have a smaller introgression. And of course, from my experience, the picture is getting clearer once you are cloning the gene, and then you can see where it's expressed, exactly which tissue.
Yeah, but you should be able to see it. I mean,
like
stay green. Do you get any phenotype for stay green?
No, they look identical, but we will try to look into it better this year.
Thank you.
What kind of libraries did you make and sequence to get that assembly with a 60 kb N50?
Okay, so the 60 kb N50 was done by Ling et al. It's a paper that was published in Nature 2013, and they use a whole genome shotgun approach. I guess you can look at the paper and see this data. I'd like to thank you.